My lab is really interested in understanding how cancer spread from one side to a distant side and form secondary tumor called metastasis. And the reason we're interested in that is because metastasis really is the main reason killed majority of the cancer patient. And however, we know very little about this process because it's very complex. It involves with a small number of cells shed from the primary site into the blood system and then carried into distant organs. And then from those cells, only a small subpopulation can eventually grow. And for many years, studies mainly focusing on the primary sites and some biopsies of metastasis. But the cells in the middle, called circulating tumor cells, those rare cells in the circulation, are really overlooked over the years. And for main reason for that is because there is a challenge in isolating those cells and in enough quantity to study the biology of those cells. So my lab really have used the ways to isolate precisely those rare cells in the cancer patient blood and try to expand those cells into a new resource that we can now study in great details about the property of the cells. So eventual goal is to understand how those cells shed into the blood and how do they gain properties like survive in the blood circulation, and eventually what are those cells, what subpopulation of the cells eventually grow in the metastatic sites. We can sample the cells from a simple blood draw, which is really less invasive than taking a biopsy from a patient tumor sites or metastatic sites. So those cells kind of really represent what patient's current tumor biology. So we have done previous studies showing that with enough of those cells expanding in culture, we can actually do the drug screen and to inform potentially in the future to inform clinicians about the best treatment options for the patient in the current stage. So right now we focus specifically on breast cancer because we actually develop majority of tools in breast cancer, but in the future we hope that we can expand in more cancer types. We hope that with enough knowledge gained, we can help, for example, provide information about predicting where the metastasis is going to be and to monitor patient more rigorously. And also we can identify what are the biomarkers, what are the driven factors that highly express in those cells, so we can tailor specific treatment options for those kind of patients. So that can really provide a way to help better treatment choices for the patient.